The overall goal of this procedure is to isolate mitochondria associated with translating mRNAs. This is accomplished by first harvesting yeast cells grown under mitochondria-enriching conditions. The second step is to gently lyse the cells in the presence of cyclohexamide. Next, mitochondria are separated from soluble components by differential centrifugation. The final step is extraction of RNA from mitochondria and control samples. Ultimately, northern analysis is used to show the purity and quality of the mitochondria-associated RNA.
The main advantage of this technique is that many types of RNA can be isolated in a single preparation. Therefore, a comparative analysis can be made.
This method can help answer key questions in cell biology, such as intracellular targeting and organelle function.
In this procedure, mitochondria will be purified from 100 to 150 milliliters of yeast cells, grown to an OD600 of 1 to 1.5 at 30 degrees Celsius on a non-fermentable growth medium. Centrifuge cells at 3,000 times gravity for 5 minutes at room temperature. Discard the supernatant and wash the pellet with double distilled water. Centrifuge again. After discarding the supernatant, weigh the cell pellet. Typically about 0.6 grams is obtained from 100 milliliters of cells. Resuspend the pellet in 1 milliliter of DTT buffer per 0.5 grams of cells. It is important to treat the cells with a reducing agent in order to break the disulfide bonds within the cell wall, thereby improving